resource wolves, working with targeted delivery of charged sodium channel blockers to produce pain selective anesthesia. And then he became a young faculty in Harvard Medical School, working with selective silencing of peripheral neurons to inhibit chronic pain and itch, mechanisms of inflammatory pain. And now he's the faculty in the School of Medicine at the University, and he's going to speak on this recent course, the somatotopically-based approach to study mechanisms of detection and transmission and perpetuation of pain. So thank you very much.
Thank you very much. Can you hear me? Good. Well, thank you very much for the introduction and for the invitation, obviously. I would like to share with you today some of the questions we are asking in my lab about how pain is detected, transmitted, and analyzed in normal and pathological conditions, especially what are the mechanisms underlying perpetuation or chronicity of pain. And instead of giving you one project that we are running in the lab, I'm planning to give today an overview of some projects. But the bottom line is that we're trying to develop a new approach. Basically, will allow us to study changes at the level of either single neurons or neuronal networks that are directly related to the activity in the periphery. And we are trying to do it by in some topic way, so we can relate stimulus or receptive field of the periphery to the specific population of neurons in the central nervous system. But I'll take you through some questions. The first obvious question is, why study pain? And I think that the answer is not very obvious. However, I must say that according to the new definition of pain, chronic pain, neuropathic pain, it's a disease of somatosensory system. So it's not anymore a sign of any other disease, secondary sign of inflammation, trauma, or some kind of injury, but it's actual disease of nervous system, like Parkinson, Alzheimer, and any other disease that's very well known. But the numbers are different. 20% of European people suffer from chronic pain. 30% of America. Okay? 17% in Israel, as you can see here. And our treatment abilities are...
How come that there's such a huge heterogeneity in countries?
It's okay. First of all, it's the only survey that's available. It's a telephone survey. It's not very well controlled, but it's the only data that the International Study of Pain have right now for Europe. Yes, it's kind of interesting that in Norway, almost 30% of people suffer from chronic pain, although in Spain only 12. I don't know, maybe it depends on the amount of sun or what they have to do, what they drink before the phone call. But okay, so maybe those numbers are not exact numbers, but if you ask clinicians, this is chronic pain, all this variety, like a migraine, chronic low back pain, fibromyalgia, and some other diseases, it's very wide distributed, and our abilities to treat it are very, very low. Okay, we're going to talk about a little bit later. It's quite low. Come on. We'll see it. Okay. Okay. So the question to be asked is what is pain, and it seems kind of easy question, but it is not. Even by looking on this picture, so this guy could be. Can we somehow reduce the light in front? Right here. Yeah. So this guy could either be suffering from cerebral migraine, or could be just miserable, as said. So pain could be either emotion or sensation. The first question is whether it's sensation at all. And if you ask Greeks, the ancient Greeks, they would say that it's just an emotion. The term pain was influenced by the name of Greek goddess Paina, the goddess of revenge. It's clear emotional relation. Aristotle, who basically defined the senses, okay, never thought that pain is one of them. He claimed that each sense could be either the level of pleasure or pain. Okay, it's clear. Pain is clear emotion. We now obviously know that pain is a sense. So the next question I'd like to answer is whether pain is specific sensation. Also not a trivial question, because it was a lot of debate about it. One of the first supporters of the pain as a specific sensation of kind of labeled line for pain was Maximilian Monfray, who basically defined a minute spot on the skin. And these spots were responsible only for specific stimuli. Some of them were responsible for mechanical stimuli, some of them were responsible for thermal stimuli, and some for pain. Kind of the origin or the first place for this first start of the labeled line. So he was an initiator of the specificity theory, meaning there's stimuli that, independent on their intensity, could be either innoxious or noxious. And pain is indeed a specific sensation. Okay, but the problem was that how pain could be specific sensation if you can feel pain as a result of thermal stimuli or mechanical stimuli or chemical stimuli. That's why the additional explanation was provided, and it was thought that maybe pain is coded as a rate, as a frequency, basically. So the theory by Earp, who claimed that there is no specific pathway for pain. There is a common pathway for noxious and noxious stimuli, and the higher the rate, it would be perceived as pain. Lower rate we perceive it as not painful sensation. Although the dilemma of various stimuli that can produce pain was solved by Charles Sherrington, who claimed that we indeed have specific labeled line for pain, and this labeled line, these neurons, are not sensing mechanical, thermal, or chemical stimuli, but they are sensing the hardness of the stimuli. Okay, and therefore he called the system a receptive system from Latin nocere, to harm. Okay, so we have a specific system that could sense harmless. Okay, but then another theory was proposed, and this theory said that maybe we do have some of the cells that are specific, but in order to feel pain or various color of pain, it must be patterned somehow. Okay, so basically it's the variety of firing of pattern of firing of neurons that will define basically whether you feel pain or what kind of pain we're going to feel. But the final, I think that from my point of view, for the system that I'm working on, the final proof that we indeed have this specific labeled line for pain was published not very long time ago, about 60 years ago, by Ed Pearl, finally recorded from a neuron, from peripheral neuron, that responded only for painful stimuli. As you can see here, this is the paper, and this is the recording from peripheral neuron cat. And when he applied not noxious or non-painful stimuli, low intensity stimuli, the neuron was silent, and the neuron started firing only when stimuli was in a noxious range. Okay. So at least part of the system, it's labeled line, it's specific. We now are able to record at much higher a resolution, and when we record from the peripheral neurons, we can see that we have not only one labeled line for pain, but many. And there are pain neurons that could respond only for thermal stimuli, only for mechanical stimuli, and neurons can respond to both. Okay? These neurons transmit information to the spinal cord. In the spinal cord, some of them transmit information to selective area for pain, the superficial areas of spinal cord. But some of them some of the pain neurons project to the deeper areas. There they converge on the transmitted cells that also receive information from non-painful neurons. Okay, so it's kind of, again, we have the same problem. We have labeled line that somehow meets and talk at the level of spinal cord. And so the latest theory was proposed in the 60s was theory from Warren Melzack, basically concluding that we may have different type of neurons, painful or non-painful, but in order to sense pain and modulate pain, it must converge at some level in the common neurons. And then rate or pattern will basically characterize what we're going to feel.
So, the last question?
Yeah.
I want to understand, if I touch the warm object, I sense that the temperature or whatever of the object,
Yep.
for example.
So if I'm, let's assume we have warm coffee and hot coffee. If I'm going to put my finger in a warm coffee and then in a hot coffee, I will sense it from different set of fibers.
And it's not simply a threshold issue.
No, no. We have totally separate set of fibers that express unique, and we're going to talk about a little bit, unique proteins that can sense only high threshold stimuli. So it's not a threshold issue, but interestingly, what this theory basically saying that if I have painful sensation right now, and if I, together with painful sensation, I can apply another stimuli, not painful stimuli, this non-painful stimuli could affect my pain, okay, which is a very well-known technique in treatment called TENS. But from an anatomical and theoretical point of view, the nociceptive system, it's a separate system from molecular and anatomical means. Okay, so then after the spinal cord, the information flows towards a thalamus and then the cortex, and then when we measure the activity obviously on large scale in cortical areas, pain activates a pattern of, or let's say it's several areas, and we can call these areas that activated by pain matrix. Okay, so there are several areas: primary somatosensory cortex, secondary somatosensory cortex, anterior cingulate cortex, prefrontal cortex. All of them are activated by acute pain, and all of them are modulated by chronic pain, or as a result of chronic pain. So we have both the changes in architecture, in connectivity, and in function of these areas in humans that suffer from chronic pain. The problem that there is no correlation between the action, between the intensity of peripheral noxious stimuli to the amount of activation of these areas. And the recent theory telling us that maybe this matrix is not sensing pain, but prominence or silence of the stimuli. So this is not very clear. And to all this unclearness about pain, we can add some other parameters. The first parameter, it's how come we can behave as something is painful, but we don't have any painful stimuli, and on the contrary, behave as if we have no pain, but we have, I hope you can see here, there's a syringe, when we do have a noxious stimuli. And how we can explain that the same stimuli, same painful stimuli can produce totally different behavioral reaction among us, as you can see here. So I'm not very good in answering the question what is pain. And since I'm not very good at it, I'm going to the literature and I'm going to use a poetic explanation. And I really like this one. I'm sorry, it's pretty upstairs. Pain is a perfect misery, the worst of evils. Okay, John Milton, Paradise Lost. Okay, so is it indeed the worst of evils? I think so. Because again, as I mentioned, there's a huge amount of symptoms that could be explained by chronic pain: migraine, fibromyalgia, chronic lower back pain, and so on. Okay, there's a huge amount of money spent, and people are suffer from day to day. Okay, and you can disagree, but I think there's no very efficient treatment. If you look on a toolkit that we have, almost no treatment are specific against pain. So we have here a anti-inflammatory, again anti-inflammatory, antidepressants, anti-inflammatory, antiarrhythmic, anticonvulsant. Only opioids are against pain. None of them are selective against pain. And maybe so basically our treatment algorithm here, it's empirical, totally empirical. So when we look on this graph, which basically explain how many people we need to treat in order to receive 50%, sorry? 50% decrease in pain intensity in only one of them, we see that in the best case scenario, with tricyclic antidepressants, we need to treat 3 patients. But when we use gold standard treatment like gabapentin, Lyrica, all the stuff that you heard about, we need to treat 5 to 6 patients in order to get improvement, 50% improvement to only one of them. And the reason for this horrible, I can say, numbers and the horrible success in treatment of pain, it's because our approach is empirical and not rational, not mechanism-based. But the problem with pain, as you will see in a second, I hope, that it's very hard to understand the mechanism of pain, because the pain occurs at multiple levels. In each level, from the periphery to spinal cord, the pain stimuli produces plastic changes. They change all the time both the phenotype of neurons, and the way the networks that deal with the stimulus works. Okay? So one way that we are doing as a pain scientist to take this pain.
Sociological effect. You have to treat 6 patients, that one of them will.
No, it's meta-analysis study. It's not sociological. It's just
One way to say percentage.
Okay. So one way to study pain, just they construct it to several compartments and see what happens in each compartment. So the first compartment will obviously be the transducer, and the transducer will detect external or internal stimuli, heat, cold, chemical stimuli, and will translate it into electrical signal. Okay? Then this electrical signal should be encoded, okay, and transferred or transmitted on the cable toward the two analyzers, two processors, one in the spinal cord, smaller one, and one in the brain. Okay? So if you want to take the more biological give a more biological explanation, so these transducer channels, these transducers are basically channels that express selectively by pain neurons, and most of them are from the family of TRP channels, transducer potential channels, that were discovered or first described by Baruch Minke. Okay, these channels respond, and actually we know little how, respond to noxious stimuli, and when they open large cation non-selective pore that produce strong depolarization that then received by sodium channels, and they encode it into rate, in action potential, in specific frequency, and then it comes to the spinal cord, and nobody knows, and we'll see what happens in the spinal cord, and then go to brain, and again, nobody knows what happened in the brain. Okay, so let's take it piece by piece. Okay, and what we can see here is the terminal of nerve ending, which is surrounded by skin cells, keratinocytes, and stimuli that affect these TRP channels. So so these channels could translate both, first of all, high threshold noxious stimuli. It could be noxious heat, noxious cold, and some endogenous stimuli, let's say ATP or pH. So when we have some trauma or our muscle work hard enough and we have some release of ATP, or our pH level are lower, we can have an activation of this terminal and we can feel pain. The question that we asked when we look on this terminal, whether only noxious stimuli or injury-induced stimuli can activate it. It will be interesting if also inflammation could be sensed directly by pain fibers. So we'll aware immediately about the inflammatory process that takes place in our body. So inflammation, which is part of tissue injury, but it could be a separate process, is described by release of variety of pro-inflammatory factors that we call inflammatory soup. Some of them are cytokines that are released by immune cells, some bradykinin, and many others. So we decided to check whether these factors could activate, similar to cold, heat, or other factors could activate nociceptors. And we started with the cytokines, and basically what we found is that nociceptors are not just nociceptors, so they not just sense harms, but they also inflammosensors, because the cytokines, at least the major one, interleukin-1 beta and TNF-alpha, directly activate them, and I'll show you some of the examples. So these are the current clamp recording from the peripheral neurons, from cell body peripheral neurons, before and after application of this case TNF-alpha, in this case IL-1 beta. So what we can see here that after application of cytokines, and actually it happens a minute after application. This is 10 minutes, but we can see the effect after 1 minute. So it's very short, very acute effect. Sorry. We can see that previously a subthreshold stimuli, okay, this one become now superthreshold. And if subthreshold stimuli here evoke only one action potential, here the stimuli evoked train of action potentials. Okay, so so IL-1 beta and TNF-alpha both activate and sensitize or produce hyperexcitability of pain neurons. And the mechanism, actually, it's very interesting. I would like to say a couple of words about the mechanism. So it could be several reasons for it. So among the reasons that we found, it was that IL-1 beta and TNF-alpha, in actually other prefrontal mediators, increase the sodium current. So as you can see here, this is the summary of recording of peak sodium current versus a voltage, and application of TNF-alpha and IL-1 beta produce increase in peak sodium current. And the mechanism is unique, at least what we know. So we would expect that the increase in sodium current could be either because of increase of the amount of channels on the membrane, or some changes in the fast properties, fast activation, fast inactivation. We were disappointed in the beginning to find out that there is none. But what we did found that instead of increasing the number of passive channels, of channels to the membrane, TNF-alpha and IL-1 beta increasing the number of available channels by affecting the process called slow inactivation. So slow inactivation is a property of sodium channels that allow basically the channel to enter inactive state from closed state. Usually channel will be open and then will be inactive, but slow inactivation will promote channel to go to inactive state even without opening. What does it mean? It means that in the resting potential, when cell receives information from the periphery, it basically operates with less than maximum amount of channels. In normal condition, it's usually 60%. So we have 40% of in slow inactive state. IL-1 beta and TNF-alpha relieve this property, relieve the slow inactivation. So now cell can operate with 80% of channels, which make it much more excitable. So and actually this is common mechanism for different cytokines. Therefore, we decided to check what will happen if we reverse this effect by applying the drug that affects slow inactivation different direction. And there's a drug, anti-epileptic drug called lacosamide, and lacosamide is doing a effect which is opposite to IL. Let's say this is normal response. IL-1 beta shifts the curve to the right, and lacosamide shifts the curve to the left. So when we applied lacosamide together with TNF-alpha, we basically were able to abolish this effect of TNF-alpha, and we're able to bring cell back to the initial normal excitable state. Okay, so to sum up, what we show here that in addition to this function as sensors of strong stimuli, nociceptive neurons can also directly sense information. So basically, there's a new way of interaction between immune cells and neuronal cells, and the mechanism is unique, and we can affect this mechanism and basically, hopefully, alleviate or improve inflammatory pain. Okay? Pro-inflammatory cytokines, in addition to this direct effect on nociceptors, have also very well known effect on sensitizing nociceptors, meaning they're not activating them directly, but they can change the threshold. So these channels that I explained earlier that respond to noxious heat, let's say this channel called TRPV1, it responds to heat about 40 degrees and up, will be activated, only when I put my finger in hot water. But during the inflammation, variety of factors of this inflammatory soup will shift this threshold toward normal temperature. Thus, in normal body temperature, this channel will be active. This channel will be active, meaning the nociceptive field, the specific path, a labeled line of pain, will be active, and we're going to feel pain. So we have this endogenous activation of TRP channels, of pain channels, and the question is whether we can use this activation, in order to block pain. And the answer is yes, and I will show it in a second. But before it, I would like to share with you the basic of this project. And we demonstrated a couple of years ago the way that we can selectively block or affect pain neurons, and we did it using this TRPV1 channel. So this one will be TRPV1 channel. TRPV1 channel. This is the channel that's selectively expressed by pain neurons. And again, it's large, cation non-selective. So what we have done, we injected, or we used charged sodium channel blocker. Sodium channel blockers, at least some of them, in order to block sodium channels, which is this one, need to act from the inside. So when we apply from the outside charged sodium channel blockers, they couldn't work because they couldn't penetrate the membrane. So when we apply this charged sodium channel blocker together with the opener of TRPV1 channel, in this case capsaicin, the ingredient of hot chili pepper, this channel was open, and this charged compound penetrates actually easily via the pore of the channel, enter the cytoplasm, and then block sodium channels. And that's what happens. So this is the recording. Okay, this is the actual recording from a peripheral sensory neurons. We have two types, small one and large one. All these lines are basically recording of sodium currents. So when we record from small cells, which are sensitive to capsaicin that activate trip channels, when applied capsaicin in QX, as you can see here, this is before capsaicin. So QX is charged sodium channel blocker. By itself, it didn't do anything because it's from outside, it can penetrate the cell. But when we co-applied with capsaicin, sodium current disappeared. When we applied capsaicin in QX on the large neurons, non-painful sensory neurons, there was no effect whatsoever. When we did the same experiment with the current clamp, now recording the ability of cells to generate action potential, we're able to totally block action potential in pain neurons by using capsaicin in QX, but non-painful neurons weren't affected at all. Okay? And behaviorally, so this is an experiment that I would like to explain to you. What we did here, we basically measured the reflex. It's called jaw opening reflex in rat. When we stimulate the teeth, the rat with painful electrical shock, the rat will open the jaw. Okay, so we can basically record the EMG from the muscle that is responsible for opening the jaw. We then applied capsaicin QX to the sensory component of the reflex. And when we apply capsaicin QX to the sensory component of the reflex disappears. Okay, but when we applied the capsaicin QX to the motor component, to the myohyoid nerve, which is responsible for the motor response, the reflex remained the same. Okay, application of lidocaine blocks everything. Okay, we produce a variety of experiments on different animals just to show you in large animals because it's much more relevant to humans. It's large animal. So application of capsaicin QX works similarly, both when we apply it nearby the nerve, using nerve block, as you can see here, lidocaine blocks everything. This is the sensory pain block and motor block. 2, it's a normal, 0, it's blocked. So when we apply capsaicin QX, when we apply lidocaine, everything was blocked. When we apply capsaicin QX, the pain was blocked, but the motor activity remains intact. Okay? And when we produce spinal anesthesia with the same combination, it was exactly the same effect. It could be relevant for epidural, it could be relevant for nerve block, it could be relevant for many, we call it surgical operational pain. Okay? But whether we can indeed use it to block inflammatory chronic pain. As I mentioned before, during the inflammatory pain, these trip channels are already open. So we don't need to use any capsaicin or any other activator. We can just apply charged sodium channel blocker and see whether we can block pain fibers. And it works. So here we're producing inflammation by injecting complete fluid adjuvant. This is the agent that produces steroid inflammation, edema, and a pain. So how we know it produces pain? This is so we measure mechanical threshold, response of rat to mechanical stimuli, and this is time. Normally, animal will respond around 16 grams. When we apply CFA, complete fluid adjuvant, the threshold drops to 4 grams. Animal become much more reactive, much more in pain. When we apply CFA with charged sodium channel blocker with QX314, the pain was totally blocked. Animal become totally irresponsible for any painful stimuli, and there was totally selective effect, meaning that we can block inflammatory pain just by applying QX314. But if we are able to do it, maybe we can use this technique not just to block pain, but also to block any other fibers or sensations or that express this kind of channels. And here it's not connected directly related to the pain topic of my talk, but I would like to show it to you because I think we use it nicely to answer some scientific questions. So the question is whether we can indeed silence specific population of neurons and then to understand their function. And our first candidate were itch neurons. So itch neurons and also peripheral neurons. Actually, nobody knows what are they. Okay, so we know that each sensation is somehow similar to pain sensation, and many people thinking that maybe those are the same neuron, and indeed we have some kind of a population code. So we can differentiate between pain and itch. Interestingly, itch producing agents or pruritogens, it could be histamine or some others, are not activated directly in the neurons. Each of them activates its own receptor. Let's say histamine activates H1 receptor. H1 receptor activates TRPV1 channel. The TRPV1 channel produces the polarization strong enough to create a sensation or action potential and sensation. So again, similarly to the inflammatory pain, during the itch, we also have endogenous activation of these channels. You can use QX in order to block it. And this is just an example of the experiment. So this is the calcium imaging experiment from peripheral neurons, cerebral peripheral neurons, and this neuron over here responded to histamine by increasing intracellular calcium. We then could record from him sodium current and see that when we apply histamine with QX, we can totally block sodium current. But when we record from another neuron, peripheral neuron, that is not responsible, does not react to histamine, application of histamine QX did nothing. And when we apply histamine QX, the itch usually evoked by histamine disappears. And the same, I'm not going to explain to you that there's also histamine independent itch induced by chloroquine. So we got exactly the same effect. Again, clinically very relevant, very interesting. Many people suffer from itch, and we can now selectively block itch neurons. But what we think that we can, again, as I mentioned, answer some more basic questions. So Ogden Nash once said that the happiness is having scratch for every itch. It basically defines what happens, but nobody knows what's itch. And so we have this interesting sensation, and people still don't know whether its peripheral or central representation are the same as in pain or are totally different. And we think that here we can at least give some idea of it. So this is our basic knowledge about itch and pain. And as you can see here in this cartoon, pain and itch are separate. So this is the skin and peripheral neurons, and this is the spinal cord. The problem is that peripheral neurons that transmit pain and itch express exactly the same proteins on their membrane. And they both respond, so when recorded both in humans and rats, from fibers that respond to pain and itch, all of them will respond to painful stimuli and to itch-induced stimuli. So nobody actually knows how we can differentiate between pain and itch. So what I decided to do, we decided to see whether if they can block all itch-transmitting fibers, whether it will affect pain at all or not. So if it will block all itch and it will affect pain somehow, meaning that pain and itch transmit by the same line. But if not, maybe they are separate. So what they have done, basically we block by applying histamine with QX and non-histaminergic agent with QX, we totally block itch. So animal will not scratch upon any of known pruritogens. And then we check what happens with the pain threshold. And nothing happens with the pain threshold. So pain was the same, and not just the pain, but all sensations were the same as before the application of itch blockade. Okay? What we can see here that we check pain threshold response for mechanical stimuli, response for thermal stimuli, cold plantar assay, response for non-painful cooling, and sticky tape tactile assay, meaning that this is the not painful sensory test. And application of histamine QX and chloroquine QX, blocking all itch sensation, did not affect these kind of behaviors. While lidocaine, it is. What we can suggest from here is that pain and itch transmitted by separate labeled lines, and there are some convergence, and we know there are some convergence in a dorsal horn. Okay? So now let's go back to pain. We talk about dorsal horn, and this is a recording from a dorsal from cell in a pain level in dorsal horn. And as you can see here, recording from this. So this is recording after peripheral stimulation. So this cell in dorsal horn basically is not not responsible for this receptive field. So when we stimulate in A1 area or B1 area, this stimulation will not affect, or will slightly affect the activity of the cell. If we then apply mustard oil or capsaicin, the substance will strongly affect or strongly activate this area. Suddenly, okay, the cell will become responsible for this field, for this area in the skin. So basically, what happens here, we have case of plasticity that we call central sensitization. So sensitization is a plasticity that occurs in spinal cord level, and basically it occurs in several ways. The first way is to have synaptic facilitation between pain neurons and central neurons. And in addition, we also have sprouting from non-painful neurons to painful neurons. So in this case, non-painful stimuli that used to produce non-painful sensation will now produce painful sensation, phenomenon we call allodynia. And the mechanism responsible, as almost all mechanisms that related to this experience-dependent plasticity, it obviously depends on calcium. This is an experiment. So this is spinal cord slice, and this is root, which is attached. And these areas, transparent areas called substantia gelatinosa, those are basically the pain areas of spinal cord. So low resolution calcium imaging from these areas, in this case before tetanic stimulation, before strong stimulation, produce some calcium influx. But after tetanic stimulation, after priming, we have much stronger calcium influx toward the cells in substantia gelatinosa. And this is the schematic explanation of what happens. And basically, some of the mechanisms are well known. This is the normal condition. In these conditions, calcium could penetrate only via NMDA channels that are basically blocked by magnesium. But after tetanic stimulation, there is insertion of new AMPA receptors that are calcium permeable, that significantly increase calcium entry and plasticity. We're also trying to understand what happens there, and so what we found out by recording from substantia gelatinosa after nerve injury, we found that if before a nerve injury all the synaptic activity was indeed mediated by AMPA receptors, and everything was blocked by CNKX, after SNI spare nerve injury is the model of chronic pain. After nerve injury, we suddenly find out that there's some additional component in the synaptic transmissions, and this component, which is much slower than AMPA, is resistant to CNKX. And actually, I'm showing this data to you not just because of the data, but also because of this slide. It's called voltage club recording from ADC. What's ADC? Any damn cell. Okay, it's real. So in pain research, we don't know what we're recording from. Okay, it's neither in the spinal cord, not in thalamus, and not in cortex. So you can see in the pain papers, people record from anterior cingulate cortex cells and see some changes in threshold and activity, and primary cortex in change activity, but nobody actually recorded from from cells that somehow directly related to the area from which pain is initiated.
Why?
Because nobody actually mapped these areas. Okay, so basically the main project in my lab, which is now only as in the level of ideas, is instead of doing deconstruction of pain and record as we used to either from the cells in the periphery or from the spinal cord slice or from brain in totally detached areas that is not connected anyhow to the periphery, to try to reconstruct pain and actually follow propagation of nervous stimuli from the periphery to the central nervous system, and try to understand the changes that are related to specific stimuli and specific location. And in order to do it, we must have the system. And in order to, so it's basically the common problem in learning and memory. So we need to find the specific population of cells that respond to specific stimuli. We can change the stimuli we can either increase the stimuli or decrease the stimuli and see how it's affect the behavior of this cell population. And a classic model for the study is a whisker barrel system. So in the whisker barrel system, we're lucky to have specific cluster of cells at each level of analysis that are responsible for specific receptive field. And those receptive fields are whiskers. Okay? So if we look on primary somatosensory cortex, there's area there that's very well organized. Okay? And we can see very clear structures of rows and columns. And it's the same structures exactly replicated on the face of the animal in the way of the whisker. And basically, each of these clusters, we call them barrels, is responsible for the information that's coming from specific whisker. And this map is recapitulated in each level, in brainstem, which there we call barrelets, in thalamus, and in primary somatosensory cortex. So my idea was, when I look in the system, is or my hope was that maybe this, sorry, and what I forgot to say is that this whisker barrel system convey mechanosensitive information about whisker deflection. And this mapping basically allows to rodent to do amazing things with this. So basically, a rodent can see with the whisker. So what rodent is doing is basically it protrude the whisker toward the environment and search with the whisker, very similar to us when we enter the room and our eyes are shut. We produce search. So this is exactly the same pattern that rodent will do with the whisker. So this mapping allows to do very high tasks. And what I hope to find is that these whiskers also innervated by pain fibers. In this case, maybe this structure could also work for pain, and so we can study pain plasticity. So I went to literature, and I found out that although this area is very strongly responsible for mechanical sensation, about 30% of fibers that innervate the follicles are pain fibers. Okay? And if you destroy these pain fibers, there was a paper by Marshall in Nature, if you destroy these pain fibers by local application of capsaicin, and the capsaicin could basically kill nerve endings, if we destroy this information from the whiskers, we destroy these nice somatotopic related receptive fields in cerebral cortex. So maybe we have some kind of connection here. Actually, when I look a little bit deeper in literature, I found out that reorganization of somatotopic maps after killing of these pain fibers occurs at each level, in trigeminal nucleus and in thalamus.
Those pain fibers are which part of the body? Like all the body?
No, those pain fibers innervate the whiskers. Only the whiskers. Yeah. So, what I'm hoping to do, and now it only depends on the equipment, I hope it basically to follow the information flow from the single whisker to identify pain-related network at the level of brainstem, the level of thalamus, and the level of the cortex. Okay? Try to identify, to somehow characterize, in some level, behavior of this network, its local network that's responsible again for the information, painful information for selective receptor fields, and then to produce very local model of either chronic pain or inflammatory pain by either killing nerve or producing very strong inflammation in selective whisker, and see how it will affect the behavior of this network. Okay, so we're just in the beginning. And we have the idea. So I will just show you one piece of data. But, so first, basically, what we need to know, we need to trace the fibers. We need to trace the fibers from the whiskers and see whether if the cells in the peripheral cells are responsible. What is the relationship between peripheral cells and the whiskers, and what cells they activate in the level of the brainstem, and then thalamus, and then cortex. So what we started to do, basically, we're now developing a new approach to trace neurons, and we call it neuronal GPS. So not the global positioning system, but neuronal positioning system. And we are doing it by injecting dyes to the periphery. So we work on this model on first on paw, and then we're going to apply it to the whiskers. We're injecting the dyes that could be uptaken by neurons. And we're injecting 4 different dyes to the same location. So this area is innervated by different neurons in the periphery. In each point, in each pixel here, will be a unique combination of dyes. We can then go back, or go up, sorry, to the cell bodies, and look on each color vesicle here. We can decipher the color combinations, and then we come back to the periphery and fit this color combination of each vesicle, and then all vesicles of the cells to the periphery. So let me show you how it looks like in real life. So in real life, this is the paw. We injected 4 different colors. I'm colorblind, but my student told me it's 4 different colors. 4 different colors to the paw. And there we're looking on at the level of a dorsal ganglion and dorsal cells, that each of them contain unique combination of vesicles, and each vesicle contain unique combination of colors. Okay, this is the trigeminal, dorsal ganglion, dorsal cells. We then take each vesicle, and again, we can know exactly what is the color combination of each vesicle, and we can build on the periphery the map and find the most fitted pixel for this vesicle. And by using different algorithms, we can find 0.5% of correlation, right? So we can basically draw the map of the receptive field, or the axonal spreading of each neuron. As example, you can see here, so this area is basically innervated by 2 cells, number 3 and number 5. Interestingly, number 3 is a small neuron, it's pain neuron, and number 5 it's large neuron, non-painful neuron. So then, what we're going to do, we're going to follow these colors toward the center and see what are the specific cells this neuron is innervating in the central neural system, at the level of trigeminal nucleus. Okay, so this is the idea. And obviously, in order to prove this whole idea, so it may work, we need to do an experiment in vivo while we can record activity of layer 2-3 or hopefully layer 4 neurons by stimulation of painful stimulation of single whisker. We're not there yet because of some technical reason, but hopefully we'll be there soon. What we are doing in the meanwhile, what we are studying in the meanwhile, it's kind of the last thing that I'm going to talk about it in very shortly. Anybody who works with pain would like to treat it and to block it. And when we look on the pain system, again in general, whether it's in the periphery, in a dorsal ganglion, or in the spinal cord, activation of these neurons produces release, external release, extracellular release of unique combination of factors. Okay, so if it's in the periphery, activation of one terminal will produce release of substance P and calcitonin-generated proteins from another part of the same cell. If it's in a dorsal ganglion cells, will also produce release from the cell body of different factors, in this case NGF. And in the spinal cord, synaptic transmission in the spinal cord of pain cells, it's not just by glutamate, but there's also a lot of other substances that are selective for pain. So what we decided to do, we decided to see whether we can produce a machine that will sense these substances and will respond to specific or selective combinations of the substances by releasing some kind of drug. Okay? And we are using model developed by my collaborator from Bar-Ilan University, who basically described in a DNA nanorobot. Okay, this is the structure that built from single-strand DNA, and by DNA staples, you can basically build 3D structure, and this looks like a suitcase. But the interesting that you have this keys, or we call it aptamer locks. These aptamer locks can sense and respond to different substances. Okay, and this is the electron microscopy representation that this is real. Okay, so we can build in one robot up to 4 aptamer locks. So basically, we can learn these robots to behave in specific logical way. So we can build a robot that we call master robot that will respond to the existence of 2 keys. We can build 2 slave robots. Each of them can respond to 1 key by releasing another key. We can create attenuator robot that by responding to 2 keys will release inhibiting substance. And by doing this, we basically can teach this robot to behave in a specific way, as you can see here. Okay, this is the real experiment, not in nerve cells, but in immune cells, that we can at least teach them how to do and, or, and so. Okay, where are we so far? Not so far away, but this is recording from neuronal cell line, and this neuronal cell line knows how to release dopamine upon activation. So we create robots that can sense dopamine, and in response to dopamine, it could release calcium channel blocker. Okay, so this is a recording of calcium signals. It's basically it's calcium imaging recording from these neurons, and we apply high potassium. High potassium will both produce calcium influx and will activate the cells, so the cells will release dopamine. So when we put the nanorobot that can sense dopamine and to release calcium channel blocker, we can see that activation of nanorobot produce significantly lower calcium influx, meaning that we'll be able to block some of its calcium channels. So basically, what we are now trying to do, we're now trying to build nanorobots that will sense the substances released in the periphery, like substance P and CGRP, and these substances could block, let's say, pain-evoked inflammation, neurogenic inflammation, or robots that can sense co-release of glutamate in substance P in the spinal cord. It could release some NMDA channel blockers, so they can block activity-dependent activation of NMDA receptors. That's it. And this is my group. I'd like to say the students, Sagi Gudes, who did all this IO1 beta TNF alpha work, Shlomo Triel, who's doing this NPS study, or the Leibovich IO1 beta work, David Robertson did all the QX inflammation and itch work, and Shailev started work on this nanorobot projects, and my collaborators from Harvard, from Austria, Ido, from Bar-Ilan University, Yoel and Baruch from here, and Peter Rear and Katerina Zimmerman from Largen, and the grant agencies that found my work. Thank you very much. Yes, insula, for example, the area, the injury to insula, the CV could make an inadequate response to painful stimulus. The people with injury to an inadequate response to pain, meaning people with lesion in insula could start be happy or laugh as a response to painful stimuli. So there's many areas that correlate emotional response and response to pain. Also in single gyrus, right?
So and the nanorobots are in the periphery, or you are?
We can basically, so So the original robots are pretty large, so they can work only from outside. They cannot enter, let's say, synaptic cleft. We now modify them and make them very, very small.
So they are. So where are you
Applying them?
applying them?
We are planning to apply. So right now we applied it to cell culture. So just the plate. But the next step was to apply to the periphery, to the skin. So what we're planning to do just a second, we're planning to first do skin nerve preparation. Skin nerve preparation is a nice preparation. We have a piece of skin and nerve attached, so we can apply painful stimuli to the skin and record directly from nerve the response to this painful stimuli. So we first will apply nanorobots to this preparation to see whether we're able to block or modify the response to the painful stimuli. We're then going to apply it to the spinal cord. We're pretty far away from in vivo because we need to know what's the viability of these robots, how fast they will be destroyed, but we're trying.
How big is a nanorobot?
So again, the original are about a 50 angstrom. We're trying to get a much, much smaller.
So what's the target size?
Nearby synaptic cleft. If you can see it nearby neurons, nearby synaptic cleft.
And the DNA there is used actually just the fabric.
Just the fabric, yeah. So the 3D structures from DNA was published long ago, and people were just puzzled with this idea. It was nice and build some stars and write their name with it. But these people at Harvard, in Douglas, actually build something useful from it.
Thank you.
Thank you.